Dr. Milton Overholser was a graduate of our medical school and a notoriously demanding educator of anatomy at MU. And if you were here for Dr. Heeb's lecture a couple of years ago, you'll know just how demanding. While he insisted on excellence from his students, his heart was always filled with compassion for those that needed him. The son of a physician, Dr. Overholser was born in Harrisonville, Missouri in 1900. He attended MU, completed the School of Medicine's 2-year curriculum in 1923, received a PhD in anatomy from New York University in 1928. After completing his medical degree in 1931 at New York University in Bellevue Hospital Medical Center, Dr. Overholser returned to MU as a faculty member in the Department of Anatomy, and he became chair of the department in 1934. For almost 4 decades, Dr. Overholser provided MU medical students with outstanding training in human anatomy. He also conducted significant research, including fundamental studies that helped lead to the use of hormones in the treatment of prostate cancer. He became a professor of emeritus of anatomy in 1965. Outside of the classroom, Dr. Overholser was an accomplished thespian, magician, and ham radio operator, and he died in April of 1989. Few have contributed so significantly to the MU School of Medicine and its students as Dr. Milton D. Overholser. And this lecture is an annual lecture that's established in his memory. And this year we are very privileged to have Dr. Dan Longo from the class of 75, well known in medicine, deputy editor of the New England Journal of Medicine, senior physician at Brigham and Williams Hospital, an investigator and former scientific director for the National Institute on Aging. After graduating from our medical school, Dr. Longo completed specialty training in internal medicine at Brigham and Williams Hospital and Harvard Medical School. He continued training in medical oncology and hematology at the NCI and completed postdoctoral research training, Laboratory of Immunology at the National Institute of Allergy and Infectious Disease. Dr. Longo became a senior investigator at the NCI in 1980 and directed its biological response modifiers programs for 11 years. He was scientific director of the National Institute on Aging from 1995 to 2010 and also served 29 years with the U.S. Public Health Service, retiring in 2006 as Assistant Surgeon General. Last year, he assumed his current positions with the New England Journal of Medicine and Brigham and Williams Hospital. Dr. Longo is an elected member of the American Society for Clinical Investigation and the Association of American Physicians, as well as a master of the American College of Physicians. He serves as an editor for Harrison's Principles of Internal Medicine and Cancer Chemotherapy and Biotherapy Principles and Practices. MU's medical school has presented him with the Outstanding Young Physician Award and the school's highest honor, the Citation of Merit. Please join me in welcoming Dr. Longo.
Thank you so much, Dr. Smith. It's a real privilege for me to be here. I note that in the interim since the break that there are fewer people in the audience. I just want to remind those of you who are here that those who left have heard me talk before. So it's conceivable that they know something you don't know. It is a real thrill for me to be back at Missouri. I had a wonderful education here. The school has always focused on medical students as the object of learning. I see Bill Trumbauer in the audience who taught me how to deliver babies, and Howard Musell isn't here, but he let me help him do some operations, and I had a lot of practical experience when I left here that has stood me in good stead. And I think all of us who went here found ourselves very well prepared for whatever life had to offer afterwards. Even if not so well known, we became better known as people began to assess our ability to do things. So I'm really grateful to the school, and it's a real privilege to be here. Oh gosh, did I lose? Look at that. We went too long. I'm sorry. Anyway, you've heard about Dr. Overholser. One thing that you may not know about his research, he was prolific in surveying the effects of hormones on various tissues, mainly in rats. But he ventured out near the end of his research career into nutritional effects on nervous system development. So all of us had probably a piece of bread at lunch, and that piece of bread was fortified with folate. And the basis of that folate food fortification was a public health study done in South Africa, which revealed that if you used fortified folate grains in food, in cereals and bread, you could prevent neural tube defects. Well, that work is directly produced by Dr. Overholser when he was doing research here. he was the first person to report that folate deficiency during development led to neural tube defects. So he was multidimensional. This is a statement from Walt Whitman that I really feel was meant for me. The fact is that everything that we've done in research that has produced some level of success has led to a more difficult question, and a greater struggle has been necessary to try to address it. The topic I'm going to talk to you about today has to do with host-tumor interactions. The very existence of a tumor seems to be a defeat of the immune system. And you might say, well, how in the world could the immune system ever be armed to fight off cancer? Well, if any of you have ever seen a patient with end-stage rheumatoid arthritis and seen the destructive power of T cells, it's not hard to imagine that if you could focus that against a cancer, it would be a benefit. But how do you do that? We also think that the immune system is important in controlling tumors that occur that we may never see. As an example, when someone has an autoimmune disease, not an autoimmune disease, an immune deficiency disease, their risk of developing a cancer goes way up. And as we've done more cancer screening, we've detected a lot more cancers at early stage that when cured with a surgical intervention have not led to an improvement in survival. And the only way to explain that is that we took out tumors that weren't going to kill you. So presumably all the tumors that are overdiagnosed by mammography and lung cancer screening are tumors that might have been, some of them at least are tumors that might have been controlled by the immune system. And it would be nice if we just could figure out how to boost that. So I'm going to talk today about many ways that the immune system and cancers interact, and mainly how the tumors defeat the immune system to exist, because those interactions also provide an avenue for therapeutic intervention to stop and reverse it. So we all don't turn into a giant lymph node every time we get exposed to a virus, because the immune system generally knows when to turn off and knows how to turn off. But very little study has gone into the factors that regulate that. We know, for example, that the antigen that elicits an immune response very often persists, but somehow or another we are able to perceive that the danger posed by that particular antigen or that invader has ended, and therefore inhibitory cytokines stop T cell proliferation. Inhibitory signals say, okay, let's tamp down. We don't need to keep fighting. And then there's an emergence of a population called T regulatory cells that also serve to damp down the immune response. Well, tumors have figured out all of these negative regulators and figured out ways of amping them up and masking their existence, the appearance of the tumor to the immune system. So how does an immune system generally recognize a tumor cell? Well, it's pretty easy. Antibodies bind directly to an antigen that it views as being foreign. The antibody itself is the effector molecule. The tighter it binds, the more effective it is. It has several ways of mediating end results. It can fix complement. It can bind to CD16 on NK cells and macrophages and cause antibody-dependent cellular cytotoxicity. Antibodies are fairly straightforward in terms of the way they see tumors. T cells are a lot more confusing. Their antigen receptor has a very low affinity for any antigen. And in general, a T cell response that you see, let's say we do a DTH response in someone who's been exposed to PPD, to tuberculosis, and they have a positive PPD, very few of the T cells in that skin reaction are PPD specific. They have many mechanisms for calling forth, it's like a fire alarm. It's like, oh gosh, one or two cells recognizes an antigen, and they don't recognize the antigen in a naked way. They recognize a processed antigen. So what happens is cells that are specialized to present antigen chop up peptides. They insert the peptide into an MHC molecule, a transplantation antigen, and it's that combination of an MHC antigen and the peptide that gets recognized by the T cell. The T cell says, whoa, something's going on here, and puts out lots of cytokines that recruit other cells that don't have the same antigen specificity, but can nonetheless be recruited to help in controlling whatever that observation is. There are other mechanisms that work as well, and we won't talk more about them. mainly because of time. But typically a normal cell presents self-peptides bound to MHC molecules. That's how T cells see them. In a tumor cell, there are a couple of ways that they can become recognized by a T cell. One is that a point mutation in a self-protein, let's say, for example, in a tumor that's driven by a mutation in RAS. That mutation is not normal. It's not a self-protein. There's no tolerance to it. But that peptide changes the shape of the RAS molecule. It becomes chronically active, and it's been demonstrated that you can immunize with a mutant RAS protein and get protection against RAS transformed tumors because it will bind to the MHC molecule and be presented to the T cells. A second thing that can happen is a point mutation in the self-peptide can create a new epitope. So instead of being a new peptide that fits into the groove, it's the same old peptide that you had before, but it's got a different conformation, and T cells can recognize that. So those are strategies that we think T cells use to recognize tumors. There are several strategies now by which tumors avoid immune recognition. So very often they have very low immunogenicity. T cells really need to bind to their targets with a variety of structures that are on the surface. And here we have this kind of bland tumor cell, and it's the enemy that's us. There's very little different about a cancer cell than a normal cell, except the regulation of its growth and its invasive properties. Very often they don't express MHC molecules. They downregulate those very early, so if they don't have MHC molecules, the T cell will never be able to recognize it. Sometimes the tumor is capable of tolerizing the immune system against it. So, for example, it typically takes 2 signals to activate a T cell, and what will happen is a tumor will block the second signal. And when you only activate the T cell with 1 signal, that's a signal, tolerate this antigen. This antigen is going to be fine. Don't bother with it. Okay? So tumors figure out how to tolerize self-T cells against cancers. Antibodies, if you make an antibody against a cell surface protein and hope that it will use its various effector mechanisms to kill, one of the things that tumor cells do is they shed that antigen. They either internalize it and they don't re-express it, so they become negative and that antibody can't see it anymore. Or, even worse, sometimes they shed it into the serum, and so the antibody you deliver intravenously never makes it to the tumor in order to bind to it and activate immune responses. I'm not going to talk so much about this complicated group right now because much of the work that we did in our lab relates to how tumor cells suppress T cell activity, and I'll spend more of the talk about that later. But the final panel here shows that there are microenvironmental things that tumors do to coat themselves, to protect themselves from T cell recognition. In addition, there's a fact I learned here as a post-sophomore fellow in pathology. Tumors have increased interstitial pressure. So what, you say? If you take a person at autopsy who has a liver met or a lung met, and you cut across that metastasis, the tumor will bulge above the level of the liver and a bulge above the level of the normal tissue. It's under pressure, all right? And as an example of what that means, here's an experimental system that was created by my colleague Brendan Curdy. He put a colon cancer, MCA38, into the flank of a black 6 mouse, and as it grew, he would stick a needle in and measure the interstitial pressure. And over time, he found that the interstitial pressure was about 24 millimeters of mercury. Normal interstitial pressure is 0 or 1 millimeter of mercury. So this is an incredible pressure head that we're trying to deliver therapy across. So not only is it difficult to treat a cancer in the first place, but it's actively pumping and trying to keep things out. Now, this is a non-Hodgkin's lymphoma that has a lower interstitial pressure, and it's easier to deliver agents to it so that it can kill. This is an animal injected with methylene blue. Methylene blue's molecular weight's about 312. So imagine a monoclonal antibody with a molecular weight of 150,000 trying to get penetration into this tumor. It's not going to happen, all right? And much of the resistance to immunotherapy that seems to make so much sense and should be so straightforward isn't because of factors that we don't take into account. Increasingly now we're figuring out the physiologic basis of this increased interstitial pressure. It's bizarre formation of blood vessels that don't work quite right. So anti-angiogenic therapy plus monoclonal antibody therapy is going to work better. All right, you have to take my word for that. The studies are underway, but you can reduce tumor interstitial pressure with various anti-angiogenic treatments, and we think that that may be a way to defeat this particular way around cancer therapy. I talked to you earlier about antigen presentation. Dendritic cells is the name of the cell type that does antigen presentation. Ralph Steinman, who just died last week, won the Nobel Prize just before he died for identifying these cells. These cells initiate T-cell immune responses. They take antigens and they process them into peptides. They put them into the groove of the MHC molecule, and that's how we respond to things. In tumor-bearing mice, in this case, and people, but this is data from mice, this is a stain for intracellular fat. So in a mouse that doesn't bear tumor, there's a little bit of fat in these dendritic cells. But in a tumor-bearing mouse, these dendritic cells are induced by some product of the tumor to increase the expression of a scavenger receptor. That scavenger receptor causes the cell to drink in lots of fat, lots of triglycerides, and it paralyzes them. So, for example, a wild-type dendritic cell will induce high levels of proliferation in a T cell. This is a curve that shows increasing fraction. This is an increasing dilution of the stimulating cells. This is the highest concentration of stimulating cells, 1 to 10. And you can see that if you throw a bunch of T cells in with these antigen-presenting cells at a 10-to-1 ratio, you get 300,000 counts of proliferation. If you use the same number of cells from a tumor-bearing animal, you get just a little over 50,000 cell proliferation. So the cells that initiate an immune response in a tumor-bearing person or a tumor-bearing animal are defective. All right, so we have trouble initiating an immune response. But I'm going to focus most of my interest on what happens to the T cells, because that's something we happen to end up working on in my laboratory. It's not that it's most important, but it's one that I worked on, and so I'm going to tell you about it. A component of tumor adaptation to the host involves escaping the host defenses. And there are mechanisms, downregulation of the MHC molecules, induction of killing of the tumor-infiltrating lymphocytes. Tumor-infiltrating lymphocytes are dead in the water. They don't work. You know, there's been a lot of hype about tumor-infiltrating lymphocytes. They don't work. All right, I'm sorry. I wish they did, but they don't. And then there's defective antigen-presenting cell function, but there are others. And we discovered this really quite by accident. I was not interested in this. I was not interested in host-tumor interactions. I'm an immunologist, but I thought, we'll figure it out. I was wrong. Because it is possible to activate T cells in a polyclonal way to mediate an immune response, we had the idea, let's do adoptive therapy of T cells that we put in culture with anti-CD3, which activates all T cells, give those to people. Within 6 hours of culturing with anti-CD3, they upregulate their IL-2 receptor. We can give IL-2. Let's give people these activated T cells, give them IL-2, expand them to huge numbers in vivo, and surely something good will happen, right? So that's the study, and that's what we did based upon animal experiments, which showed that when we did this in a model of hepatic metastases, the more cells we gave that were activated this way, the better the anti-tumor effect. So we thought, okay, we're onto something here. Let's do this in people. So this is a peripheral white count from a patient with metastatic melanoma who we phoresed on day 1. We activated the cells for 6 hours in vitro. We gave the cells back to the patient and then gave them several days of IL-2 therapy. And this is the white count in thousands. Don't be alarmed. These were normal T cells, so a 40,000 white count was better than we expected to get, but it wasn't dangerous to the patient. We had one patient who had 104,000. The average patient had between 30 and 40,000, which is why I've shown this particular patient. We did this for 2 cycles. So while we got more in vivo expansion of these activated T cells than we even anticipated. All right, so what happened? Well, this is a melanoma lesion in a patient who we started with. You can see very few lymphocytes scattered here and there. But when we stain for T cells in a responding lesion, we're biopsying a lesion now that's regressing under the influence of our therapy. Wow, there are T cells everywhere. wow, we got it, we did it. Okay, this is going to be great. So we look at a CT scan, and this is before and this is after treatment. Okay, all so far so good. If anybody ever shows you an anecdote like this about a cancer therapy, the treatment does not work. Okay? When a treatment works, they show survival curves. This is Hodgkin's disease treated with MOP chemotherapy versus radiation therapy. Okay? So just for your own welfare going forward, don't buy it. If somebody shows you an anecdote, it probably doesn't work. And ours was no different. So we wondered, well, gosh, we got immunologic effects beyond our wildest dreams. Why didn't it work? Well, we thought back about the animal experiments, and the animal experiments we did, we started with a normal mouse as the donor of the T cells. We took normal mouse spleen out, and we gave it to a tumor-bearing animal, and the tumors melted away. But that's not what we do in patients. In patients, we take T cells from tumor-bearing patients. So we redid the animal experiments with tumor-bearing animals as the donors, and lo and behold, those who had had tumors for 2 weeks had no anti-tumor activity. Something's wrong with those T cells. All right? So being scientists, we hope, that are well-trained, we thought we'd take a systematic approach to this. What's wrong with the T cell? Is it that this is the antigen recognition complex? Note that it has multiple polypeptides that are synthesized together. On the surface of the membrane, they sort of form a channel. And in this channel, the peptide and MHC combination fit and then trigger signal transduction into the cell. The key molecule in all this is the Zeta chain. It's made in limiting amounts. All the other chains are made in excess amounts. And it's the Zeta chain that's responsible for the stoichiometric assembly of 1 gamma, 1 delta, 2 epsilon, and 2 pairs of alpha-beta chains. If you don't have a Zeta chain, you can't assemble this receptor. All right? So we asked the question, is there something wrong with the T cell receptor in these tumor-bearing animals? And looking at the anti-CD3 staining, no, plenty of T cell receptor on the surface. But when we signaled the cells with the antibody, and the very first thing that happens within seconds is the release of intracellular stores of calcium. That's just one of the first steps in T cell activation. So what we see is that normal cells activate normally, but T cells from tumor-bearing animals are very sluggish, and they never reach the normal level. They're not responding to activation signals in the normal way. So why is that? Well, we looked at the pattern of protein phosphorylation as a consequence of activation. That's the second step in the activation of cells, is that they have to activate kinases that then trigger signal transduction pathways that activate transcription factors. The transcription factors go to the nucleus and activate a panel of other genes to help respond to whatever the stimulus is. And what you can see is that whereas normal T cells, when you activate them, have this bolus of increased phosphorylation of proteins that correspond roughly to LCK and FIN, there's no such activation in T cells from tumor-bearing animals. So if we look specifically for FIN and LCK, and tumor-bearing animals don't have it. All right, what happened to it? Well, we looked at the structure of the receptor. This is a complicated experiment, and not everybody needs to care about it so much. But a way to look at the various components of the T cell receptor is to electrophoresis it in one direction under reducing conditions, and in the other direction under non-reducing conditions. And what happens under non-reducing conditions is that complexes stay together, and under reducing conditions, the complexes pull apart. So the ones that are not disulfide linked stay on the diagonal, and the disulfide linked one, which is Zeta and a key one, comes off the diagonal, and you can see it clearly. T cells from tumor-bearing animals, no Zeta chain, very little gamma, and instead this little faint blush, which is a Zeta family member that's a part of the Fc receptor on a different class of cells, has substituted and done a very poor job of assembling this receptor. So T cells from tumor-bearing people and animals don't have Zeta chain. If you don't have Zeta chain, you can't make this. If you don't make this, you can't activate the T cell. So what's the mechanism? This is a messenger RNA looking at the Zeta chain message in tumor-bearing animals, and it's not because they're not transcribing the gene. It's not that they're not trying, but in other experiments that I won't take time to show you, there's an increase in protease activity that degrades the zeta chain prematurely. Its half-life in the cell under some influence of the tumor is very short. That protease also has other effects on the transcription factors. If you can't signal, you can't cause this. I'm not having a lot of success with the pointer, but suffice it, I'll walk over here. CREL and P65 are members of the NF-kappa B family that are really important for activating lymphocytes, and neither of them are activated in T cells from tumor-bearing animals. There is another member of the family called P50, and it's truncated to P48, and it's been shown subsequently that P50 dimers, homodimers, typically the way this transcription factor works is P65 and CREL, one of them combines with a P50, and that's what turns on the gene. If you have two P50s, it turns off the gene. All right. So what seems to be set up is the tumor is causing a suppression of the activation profile of the T cell. And if you look at the production of cytokines, instead of producing the Th1 type cytokine interferon gamma, you produce much more IL-4, which is a Th2 type. We'll talk about these different subtypes in just a second. But suffice it to say that the tumor is distracting the T cells from producing things that would kill it to producing things that kill worms. And they're not worms. So it's really kind of a clever deception that they mediate. When we looked for how frequent this problem was, this zeta chain loss in cancer patients, it ranged between a low of 43% up to a high of 90% in renal cell cancer. So it's very common in patients with metastatic cancer that their T cells don't work. If you look at patients with melanoma over a larger series who are treated with immunotherapy, IL-2 does work in a subset of patients. All of the people who are long-term responders have T cells that bear zeta chain. All of the ones that don't have zeta chain are short-term survivors. We went so far as to define that it was a soluble product of the tumor that was smaller than 100,000 in molecular weight that did this, but we haven't been able to characterize what the factor is, and it may be more than one factor. If you grow a tumor in hollow fibers in the peritoneal cavity of mice so that the tumor grows but it doesn't invade and it doesn't spread and it doesn't kill, but things it produces can leak out of the hollow fibers, what you find is that it's just the same as if it was a tumor-bearing animal. So something the tumor is producing is doing this damage to the T cells. We just haven't defined what it is yet, but we will. We'll figure it out. So in summary, there are functional T cell defects. They produce structural changes in the T cell receptor that produces significant downstream alterations in signal transduction. You do not activate CREL and NF-kappa B P65, which is essential for the T cell to do what it's designed to do. There's a loss of this Th1 pattern of nuclear binding proteins, and there may be a clinical correlation with these changes in the lymphocytes. We have occasional patients in whom we've given successful therapy, and when we do that, the T cells come back normal. And it's mediated, at least in part, by soluble factors. Some of us think that this may be that the T cell desensitizes itself to overstimulation. It sees the tumor, but it's not having very good effects against it. The tumor is producing stuff to put it to sleep, and it punches out. Okay, now this is in immunospeak. One of the things, when I was in medical school here and I made the mistake of expressing to one of my professors that I was really interested in immunology, he said, "What? Immunology? Those people make it up as they go along." You know? And it's pretty much true. You know, I have to say, they had it exactly right. What are we up to? IL-39, and we have all these arrows that go here and go here. What I want to tell you about is that CD4 cells are really important, and you know that because patients with HIV have a myriad of clinical disasters because they don't have it. But naive CD4 cells have four fates, and they become different cells, different kinds of effector cells, based upon the situation they are trying to fight. And Th1, that's really good for fighting intracellular pathogens and tumors. Th2, it's great for fighting worm infections. This Th17 is very good at attacking extracellular pathogens. And then there's this group down here called Tregs. They're FOXP positive. They express the IL-2 receptor, and they can be activated to expand with IL-2. Now, didn't I just tell you that we were using IL-2 as an anti-tumor agent? It's one of those, oh, if only I'd known about Tregs. We use IL-2, and it expands Tregs. Tregs turn off the immune system. So it's little wonder that the response rate to IL-2 in serious diseases is 10% or less. All right? We are so dumb. We need to figure out how to use this stuff in a way that's more physiologic and more responsive to what the host-tumor interaction is telling us. So what we've learned is that tumors are very smart, and they make a lot of TGF-beta, and they this is a sort of cartoon to illustrate the point. Under steady state, regulatory T cells, which are the negative effectors, and effector T cells, which are the positive effectors, are in rough balance. What the tumor does is it causes an expansion of these T regulatory cells. You get fewer of the effector cells, and of course, fewer effector cells mean you get a poorer immune response. In order to get a productive immune response, you have to keep the Tregs under control. But there are times when a productive immune response is too much. Graft versus host disease is bad, right? There are autoimmune diseases, multiple sclerosis, and rheumatoid arthritis. There's a whole cottage industry now in using low-dose IL-2 to upregulate and expand the T regulatory population so that you rebalance the immune system and don't get as much autoaggression from those T cells. We're about to publish a paper in the New England Journal that you're sworn to secrecy about. Chronic graft versus host disease is a terrible disease. Low-dose IL-2 reverses almost all of the symptoms in at least half the patients it's been tried in so far. So the future looks a little bit more rosy. Again, so let's summarize. Tumor cells, bad. They make a lot of immunosuppressive cytokines. They upregulate immunosuppressive immune cells, especially T regulatory cells. We haven't talked about the myeloid suppressor cells that expand in the face of tumor cells. And again, our little contribution to this is the signaling disruption in lymphocytes as a consequence of bearing tumor. I haven't talked about all of the mechanisms. I've talked about the bottom half, but I want to pay attention here for a second to this top half. What some tumor cells do is they express these molecules CD86 and CD80 that activate CTLA-4. I think I told you before that CTLA-4 is a negative regulatory signal. It's one of the things that gets activated when enough has been enough, and it's time to turn off the T cells. And that's why ipilimumab, a monoclonal antibody against CTLA-4 that blocks it from seeing its signal, looks like it boosts immunity and has become a therapy for metastatic melanoma. There are other interesting things that tumors do. They express Fas ligand. Fas ligand will kill a tumor cell because they express Fas. So will kill a lymphocyte. Fas ligand on a tumor cell will kill through this Fas-induced apoptosis effector cells that come to fight the battle. So there are so many things that the tumors do to escape the effects of immune activation. What are we doing to try to overcome this? We are blocking Treg expansion. There are ways of interfering with their biology. Again, they're all experimental at this point, and we've done a better job of expanding them in autoimmune disease than we have in blocking them in cancer, but that's the next step. You can block the inhibitory signaling, for example, in the CTLA-4 receptor with ipilimumab. You can block the dendritic cell scavenger receptor and maybe preserve antigen-presenting function. That might help the immune response. There are several ways of enhancing antibody-based anti-tumor effects, and there's a whole series of manipulations of gene therapy type manipulations to make T-cell recognition of tumor MHC independent. So instead of worrying about whether MHC is expressed by the tumor or not, instead of worrying about whether there's a peptide that can be presented by the MHC, there are things that are on the surface of tumors that we know. We know CD20 and CD19 are on B-cell lymphomas. we can make a new receptor that looks a lot like a T-cell receptor in terms of its signaling, but its binding capacity is an antibody. And there's been recent progress in that I'll just review briefly. Am I over time yet? Okay. And, of course, reversing or preventing T-cell suppression. So this is complicated, and I don't want to go into this. Recaps what I have just said about physiologic T-cell recognition. MHC binds to a peptide, and that's signal 1, and it acts through the T-cell receptor. Again, in the tumor-bearing state, this Zeta chain doesn't work so well, so that doesn't work. In addition, CD80 or CD86 on the antigen-presenting cell delivers signal 2, and you need that for activation. But, again, tumors figure out, let's block signal 2, and you end up with tolerance rather than activation. So what people have begun to do with these chimeric antigen receptors for T cells is engineer an antibody binding site, for example, an antibody to CD19, hook it up to the signaling machinery of the T cell, and put it into T cells and see what they do. Now, the first generation cells were completely dependent upon exogenous IL-2 added to cause in vivo expansion, because it turns out that signaling just through the Zeta chain alone is pretty wimpy. By contrast, now people are adding more signaling molecules to the intracellular part, so they're keeping the antibody that binds to the antigen, the membrane-bound antigen, and they're putting much more oomph behind the intracellular activation part. So they're using co-stimulatory domains from CD28 and 41BB. These are co-stimulatory receptors, and they have additive signals and maybe even synergistic signals in some case to cause in vivo expansion. And then we just published a paper in the New England Journal from Carl June's group using this kind of receptor, in which 3 patients with refractory CLL, all 3 got a complete response, and one of them got a rapid tumor lysis syndrome from T cells that are given in this way. So that's highly promising. Of course, it's still early, and one thing you learn in this business is that around every corner there's another barrier that has to be climbed. So we don't want to get too enthusiastic about it, but I hope I have made the point that analysis of these interactions has defined for us new targets. And when we define a new target, we can find ways around it. And so the hope is still there that we'll be able to figure out how to activate the immune system to fight cancer. Thank you.
Do we have any questions? I'm sure we'll stand for a few.
Yeah. Sure.
Dan, I wonder if you could comment on the fact that cancer incidence increases for most cancers over time, and many have said that's due to the opportunity for mutagenic events, exposure to environmental agents, propensity, whatever, over long periods of time. And so you see cancer peaking in the elderly for the most part.
Yes.
What I've done in kids, mostly in the elderly. Do you have studies of normal aging of the immune system and indicate any role potentially for changes that occur with aging in normal immunity that might also bear on the incidence of cancer peaking in the elderly?
Absolutely. So the question is, are there things that are wrong with the immune system of older people that leads to or abets the increase in cancer incidence? And that's certainly the case. We don't know all of the abnormalities of the aging immune system. There are some things we know are not a problem. So you've heard about telomerase. Lymphocytes express telomerase, and that helps maintain their telomeres at a physiologic length to permit cell division. But I'm not sure that the theory has been that as you get older, you don't express as much telomerase, the telomeres get shorter, and then the cells can't divide. That's all based upon population-based studies where you take a bunch of 30-year-olds and measure their telomeres and a bunch of 80-year-olds and measure their telomere length. And in general, the average is a little bit lower. At the NIA, my colleague Nan Ping Wing and I measured telomere length longitudinally over a 12-year period in 124 people of varying ages. But I can tell you that over a 10-year period, a 12-year period, the telomere length is absolutely stable, all right, in an individual. There may be average differences in population, but the difference in the function of the immune system is not telomere length. The second avenue that we're pursuing is that metabolically things change a lot as you get older. There's more dependence in the mitochondrial generation of ATP through glycolysis and not through the Krebs cycle. And so what we are looking at is that it looks like the cell's internal system for scavenging adenosine is not as strong. And it's too complicated to go into now, and the data are not completely well formed. But older people's lymphocytes have higher levels of adenosine, and adenosine does interfere with their function. It's T cells that are the main problem. You can get B cells to work perfectly well in most cases. Where that's not the case is with polysaccharide antigens that are T independent. Older people do not respond to pneumococcal vaccine. I chuckle a little at the hard work and the pressure that's put on doctors to give older people pneumococcal vaccine. There is not a shred of evidence that is protective, not a shred. Okay, that's a bunch of people sitting in a room saying, we think this is going to work, all right, and it's a good thing to do. Let's go ahead. It probably doesn't hurt people, but the percentage of older people who respond to the vaccine is very, very small. Yes?
I'm often asked, Doc, is there some kind of test that will tell me whether I have cancer? Is the work being done of an office-based blood test that would signal that your immune system is acting normally and that there's a high likelihood that you do not have tumor activity going on because your immune system seems at rest?
Mm-hmm. There are a lot of ways of assessing immune activation. This high sensitivity C-reactive protein is one that's being highly touted, especially for vascular health. The problem is that it's highly complex, and there are multiple ways that the immune system can be activated. In general, older people have more immune activation. And although a lot of people are interested in this, it would be wonderful to be able to have a diagnostic test that would say, look, you're really cancer-free and don't worry about it. But there is not such a test now. And I think increasingly as we do more and more screening of normal risk populations, we're finding that the expense of finding the very rare person who has a tumor is prohibitively expensive. None of our tools are cost effective. You know, we just published this lung cancer screening study, and we found that lung cancer mortality was reduced by 20% by 3 annual chest CT scans in high-risk patients. But they detected excess cancers, and the overall mortality of people who were smokers, they die of heart disease, they die of lung disease. And so the overall mortality was affected almost not at all. You know, we just changed the modus of death. We didn't even change the time of death, you know, with that intervention. There's all this argument now about PSA levels and how many normal men are hurt by having a very serious operation to control a tumor that would never kill them. We don't know enough to distinguish the prostate cancers that are really nasty from the ones that you can live with and they didn't need to be detected in the first place. And there are a lot of cancers like that. I was surprised, but increasingly, as we screen more and more, we find many more cancers detected than end up ever killing anybody. Yeah?
Hey, Dr. Longo, would you be kind enough to comment on chemotherapy, since you talked so much about melanoma, chemotherapeutic changes in the last several years, innovations, whatever, new drugs available, and the impact it might have on malignant melanoma?
Well, you know, the picture for malignant melanoma is changing quite rapidly. There is ipilimumab, which seems to have an effect in a small subset of patients. It does increase median survival of patients with metastatic disease. in the 60% of people who have BRAF mutations, in particular this V600E mutation that's common, there are specific inhibitors of BRAF that have produced very high response rates. And they have made an impact compared to therapy that we know doesn't work. DTIC, I was not really happy. I'm interested in Dr. Boyce's opinion of the study that randomized people between the BRAF inhibitor and DTIC. We have 40 years of clinical experience with DTIC. We know that drug doesn't work. It's the best available, but it doesn't work. Why were people subjected to a randomization between a drug that produces responses in 75% of patients and a drug that causes responses in 10% of patients? So I think our system has lost track of the patient in some of these drug development things. From my point of view, equipoise was lost, but long before they did that study, and they should not have done that study. I would be very unhappy if somebody I knew or cared about randomized a DTIC. We know it doesn't work. It works, this drug works, but resistance emerges. We've just begun to figure out that about a third of patients will develop a second skin cancer that's a squamous cell carcinoma. And the way that happens is that when you block this pathway, another gets activated, and it causes another tumor. Fortunately, all of those have been localized and easily resectable, and it's now clear that if you add the BRAF inhibitor with an inhibitor of the MEK pathway, that you get both types of cancer are controlled. What we don't know is how long they're going to be controlled. And resistance emerges. Cancers have mutational properties that allow them ways around every one of our clever blockades. And it'll probably be a multi-layered attack that produces significant advantages. Yeah.
Medical question about Rothschild's vaccine. Then you said vaccine. So that vaccine, you take the tumor, you make a booklet bodies, and you put that with the dendritic cell, and the dendritic cell will vaccinate against the tumor, and you don't need to know what
The antigen is, right. Right.
Isn't that's one of the things where now most of cancer vaccines is headed?
Well, the cancer vaccine as a field is very old. It's very ancient. People have been giving mixtures of cells for 30 years. When I was in training at the NCI, we participated in a study where we took 3 different melanoma cell lines and used it as adjuvant therapy. And, 1 or 2 patients would get really dramatic prolongation of their disease-free survival, but it was never clear and never reproducible, and a couple of them became albinos, because of the wiping out all of their pigmented cells. I think that there are multiple schools of thought about vaccines, and progress is being made on many fronts. I don't know what the ultimate result is going to be. I think that the dendritic cell vaccine is very interesting, but one has to pay attention to the starting material. If you start with dendritic cells from a cancer-bearing person, you have to be sure that they're functional. And what works in animals, of course, when we use normal animals as the donor of the cells, is not necessarily translatable when a tumor-bearing patient. It didn't work. I'm sorry, Yeah. Well, I don't know whether that anecdote is telling us about applicability to other cases. I hope it does. I hope it's the way forward. But I'm not a betting man. I need to see the data. I'm not prepared to say this is going to be the way. I think it's going to have to be a multi-pronged attack, and it may well be that dendritic cell vaccines are among the most effective. We'll have to see.
And so the reason I asked the question is, so the Zeta chain, the CREM downregulation, are they for both CD8 and CD4, or
Yes,
across the,
both.
and they don't do that on Tregs?
No.
How do exercise and nutrition affect tumor immunity?
It's a very good question, and it's under study. The question is, how do exercise and good nutrition affect tumor immunity? It's an understudied question. We always give advice fairly glibly about, stay active, eat right, but the the amount of actual experimental data that says that that makes a difference is vanishingly small. It's just very hard to do those studies. Nutrition studies in general are very hard to do, very hard. It's hard to control it. It's hard to get accurate representations of what people really eat. I mean, they keep diaries, but it's not perfect. And again, the composition of diets are so complicated that we may not know what the active component is. For example, people said for a long time that you need to take alpha tocopherol as the form of vitamin E. But you know that makes lung cancer worse. And the natural form of vitamin E that's more prevalent, 2/3 of vitamin E in the real world is gamma tocopherol. And gamma tocopherol has nitrogen free radical scavenging ability that alpha doesn't have. And it may well be that the foods that we call rich in vitamin E are working because of gamma and not alpha. So the general nutrition centers are feeding people the wrong form. We don't know, and it's very unclear. It's very foggy right now, and it's complicated by the fact that it's a multi-billion-dollar industry, and that because they're nutritional supplements and they're not regulated by the FDA, there's a huge amount of money made, and many of the people who are making the money don't want an answer to a scientific question. So I have to tell you that as a practicing doctor, I recommend strongly, eat moderately, exercise every day, but I don't know if that has a basis in fact. Yes, sir.
I just learned about the increase in interstitial pressure in solid tumors, and I'm curious, why doesn't that choke off the blood supply?
The blood supply of a tumor is chaotic. It's very interesting that the formation of blood vessels normally follows the Fibonacci, equation. It's highly directed, and we don't understand that either. But not in a tumor. In a tumor, it'll form a ball, and very often the vessels are incompletely formed, and tumor cells themselves are part of the wall of the blood channel. We don't have any idea, but you can't really compress a tumor cell, and so we think that, even though hypoxic cells are in the center, somehow or another they get adequate supply not to die entirely. I wish they would die. Actually, it almost forms a selective environment for their survival. I think that the cells in the center of a tumor develop ways of repairing damage that are not present on the periphery. And to some extent, that's the basis of their radiation resistance, without oxygen. Yeah. It's a war of attrition. Thank you. I'd like to be like Oprah and say, for all of you who stayed, there's a free car. But
well, thank you very much, Dan. We appreciate you coming and talking and answering questions. And let's adjourn until 6:30 this evening. We'll see you at the Holiday Inn Executive Center. Thank you very much.
Thank you.
Wow, that was
great to see you.